So today I'm going to be talking about DNA replication. And first let's start with talking about DNA and its major functions. So the two major things that DNA does is first it transfers genetic information from generation to generation. And second, it regulates all the processes of your body. For example, transcription, translation, meiosis, mitosis, everything happens because of DNA replication. This is also why it's absolutely necessary for DNA to replicate. If DNA never replicated, meiosis and mitosis would slowly halve the size of the genome until each cell would die, and that would probably not take long. Therefore, it's important that DNA doubles itself to account for the cells splitting during mitosis and meiosis. For example, if DNA replication didn't occur, if we had 46 chromosomes, every generation would have less and less. And the next generation maybe would have half as much and half as much and half as much. This is also why DNA replication must be precise, because each time a cell divides, the two resulting daughter cells must contain exactly the same genetic information, or DNA, as the parent cell. Therefore, to accomplish this, each strand of existing DNA has to act as a template for replication and has to be exactly the same. So firstly, let's talk about what DNA is made of exactly. DNA is made up of nucleotides, which are the basic structural unit of DNA. And nucleotides are comprised of three principal components. These components are a pentose sugar, phosphate group, and nitrogenous bases, and a nitrogenous base. And nitrogenous bases are nucleic acids that are composed of a combination of 5 main ones in DNA. These 5 main nitrogenous bases in DNA are guanine, adenine, cytosine, and thymine. So the nucleotides basically relate to the complementary base pairing as well, because guanine always pairs with cytosine and adenine always pairs with thymine. And that's why in DNA replication, when the template strand is being replicated and additional base pairs are being added, that's the order that they're usually added in. So I mentioned parent template and original strand. Now in this image, we can see that the parent template is this strand going off right here, the blue strand. And the same here, the blue strand here is the parent template and the original template. Now the daughter and the new strand are the orange ones that are being added on due to complementary base pairing. So the new nucleotides are usually added in a 5 to 3 direction, so going this way and this way. So what this means is that one of the strands is oriented in the 3' to 5' direction. So from here, that's the leading strand. And then the other strand, as we can see, is oriented in the 5' to 3' direction, which is this one, the lagging strand. And so as a result of their different orientations, the two strands are replicated differently. And we can also see that they we can also see that here the leading strand is replicating towards the replication fork, which is this right here, and the lagging strand is replicating away from the replication fork, which is here. And so moving on, the idea of the leading versus the lagging strand is that basically because DNA is anti-parallel, the the enzyme that adds the nucleotides, which we will get to later, must move in opposite directions on both the strands. Therefore, the leading strand is when the enzyme is moving towards the replication fork, and therefore that's called copying continuously. And then the lagging strand is when the DNA polymerase is replicating away from the replication fork, meaning that it is copying discontinuously. And so Okazaki fragments are basically short sequences of DNA nucleotides, which are they occur because the lagging strand is being synthesized discontinuously. But later an enzyme will come to fill that up. Now let's go on to the enzymes that move on to the enzymes that help make all of this happen, because that's one of the most important things. So firstly, we have helicase, and the main function, which we can see here, helicase, the main function of helicase is basically to unwind and separate the double-stranded DNA by breaking the hydrogen bonds between the base pairs. So that's what that basically does. And then the RNA primase, as we can see here, its main function is to provide an initiation point for the synthesis of the DNA, almost like a starting point. And the DNA gyrase, which isn't on this picture, but the DNA gyrase basically reduces the torsal tension created by the unwinding of the DNA by helicase. So basically, it does this by relaxing the supercoils that would otherwise form during the unwinding of it. And then we have single-stranded binding proteins over here, and those basically prevent the strands from re-annealing. So these proteins help to prevent the single-stranded DNA from being digested by nucleuses, and they will eventually be dislodged from the strand when a new complementary strand is synthesized. But we also have the DNA polymerase 3, which is one of the most important main functional enzymes. And the DNA polymerase 3 basically adds on free nucleotides opposite their complementary base pairing, as we talked about before, A to T, G to C. It attaches to the 3' end, and so it adds in a 5 to 3' direction, as it's adding more to the 3' end. And so then we have DNA polymerase 1, which as we talked about, as the lagging strand is synthesized in strong fragments, it has multiple RNA primers along its length. So what DNA polymerase 1 does is that it removes those primers from the lagging strand, for example here, and then it replaces them with DNA nucleotides. And then the DNA ligase it basically joins the Okazaki fragments. As I said, that one of the enzymes will come and fix that, and that's basically DNA ligase. It joins the fragments together and it forms a continuous strand. And yeah, and so the last one here, we can see the DNA topoisomerase. The main function of that is to basically participate in the overwinding or underwinding of DNA. So the winding problem of DNA usually comes up during the fact that from here we can see that it's intertwined. The double helical structure is intertwined. So during DNA replication and transcription, DNA becomes overwound ahead of a replication fork right here. So yeah, that's basically everything. Just to go over semi-conservative replication once, what that basically means is that whenever DNA is replicated, at least one of the strands is original, so it's from the parent strand. As we can see here, that would be the blue one, and the new strand would be the orange one. So semi-conservative replication basically means that one of the strands has to be new, has to be the ones that's synthesized, used through DNA polymerase 3. But it has to be new, but one of them also has to be from the original one. So that's basically the meaning of semi-conservative replication. So yeah, that's everything. Thank you.